Well, the speakers this morning were very nice to me because everybody was talking about the palatability of the necessity for highly palatable replacements for fish meal and fish oil, and that's the topic of the next session, is the use of palatants to increase the efficiency of alternative feeds. And our first speaker today is Dr. Dellinger, whose expertise includes attractant molecules and chemistry. He's the founder of Kepley Biosystems Incorporated, which is an early stage startup enterprise designing novel olfactory technologies. Thank you.
Good afternoon. Thanks, everyone. So, as you mentioned, my name is Anthony Dellinger, and that actually says nanoscience there. It doesn't say aquaculture, marine biology. My background's actually in fullerenes, and if you do not know what a fullerene is, they're very small molecules. You can fit about 100,000 of them across the diameter of a human hair. So I was riddled with working with the unseen, or at least the difficult to see. I'm going to talk a little bit today about what we're doing, but I always like to start with why we're doing it. And we really wanted to address the hard-working lives of the fishing industry. So our technology starts as a solution for the wild capture industry, in particular crustaceans. And this industry is subject to a lot of variables. It can be labor-related, weather-related, fuel-related, location, season. But what really keeps these fishing participants up at night is bait, and that's bait cost and bait availability. Secondarily, as we all know, there's a lot of hands in this forage fish cookie jar now, and the fishing industry was suffering greatly because they do use these exact same fish to bait the traps that they use. So our approach, we were gracious enough to receive funding from the National Science Foundation in terms of a phase 1 and a phase 2, as well as a couple supplementary grants, the TECP, and the North Carolina Sea Grant Agency funded us to initially start our research. And what we did was we went out and talked to all the fishing participants and figured out what they were using as bait. We then simulated those exact same bait sources in the lab and looked at all the molecules that were coming off of them. Once we were able to collect them, we then began doing what we as nanoscientists know best is characterization. So we characterized everything that was coming off of these fish. We were able to develop a portfolio of different molecules, whether they were amino acids coming off of the fish or different decomposition products that were breaking down from the fish using mass spec and HPLC to characterize the different molecules. And in my field, I started with learning how molecules react with cells. So we wanted to see how these individual molecules that were coming off of the fish that were used as bait interacted with the olfactory behaviors or the chemosensory behaviors of the crustaceans that were being used in the fishing industry. And in doing so, we created organobait, which is what we call a surrogate fish. And had a nanoscientist developed this, it would have been all wrong. But the National Science Foundation encouraged us to get outside of the lab and go meet with customers. So we talked to a couple hundred different fishing participants and figured out what their real pain points were and where we could be helpful. They said they needed something that was clean and easy to use, something that would be a drop-in technology, something that didn't spoil, something that could be stored in ambient temperatures, was affordable, available, and effective. We started testing in 2014, mostly along the East Coast. We've worked with stone crabs, blue crabs, spiny lobsters, and American lobsters, as well as crawfish in Louisiana. We have solutions for both the recreational segments of the fishing industry as well as the commercial segments. We received funding to build our first module in 2017 at the end of the year, and we were originally making these by hand in very small quantities for research purposes. With this module, using all off-of-the-shelf equipment, that was readily available that we could incorporate into our manufacturing process. We're in the final stages of creating this first module that can generate about 4,000 baits per hour. And we believe we can improve upon this greatly as time goes on. In terms of connecting with the industry, like I said, we went out and spoke with folks that were in the fishing industry and learned what their pain points were. But we didn't really want to test our bait's efficacy in the lab because there wasn't a lot of value associated with that. We know that crustaceans behave very differently in captivity, and so we wanted to create a product from the start that would translate well into the industry. So we leveraged all of our experimental baits in the field with real fishing participants to collect our data. There were some challenges associated with this, and I'm sure everyone here that's in the aquafeed manufacturing industry, there's some psychological component with transferring new technologies into a industry that has a lot of lore. And in fishing, wild fishing in particular, the way that their grandparents did it was the right way. So it was a real challenge at first. But what we found was timing is key with any innovation. So with respect to the forage fish situation, the people that were out there fishing either couldn't obtain bait, therefore they couldn't go out and fish, or the bait became increasingly expensive, cutting into their already razor-thin margins. So due to the extension of forage fish across multiple industries, we were able to leverage that as a way to get into the fishing industry and kind of obviate some of this lore. We also co-developed it with them. We wanted to include them. We felt inclusion was very important, have their input along the way. They would essentially become our first early adopters, so we wanted to build these established relationships. Probably the hardest part about the whole entire thing was not finding the molecules that caused attraction, but creating a matrix that would dissolve and fit all of the different fishing environments that were out there, which varies tremendously from stone crabs in Florida at 15 days to Maine lobsters or in Nova Scotia where they might fish for a day. And the temperatures are a lot different as well as the depths. So we needed to make something that could be timed, kinetically release these molecules of attraction on a fishery by fisheries basis so that they were maximizing their potential. And that took us almost 2 years to do, and the NSF gave us the phase 2 to finalize our formulations, which we've came up with, and some of this data is here in terms of how well it was able to dissolve in respect to the different fishing environments that we were testing in. And then really one of the third real challenges is reliability and accuracy. So I said we focused on the East Coast predominantly, and that was so we could build face-to-face relationships and create a meaningful connection with these folks. Risk-averse participants may not put an untested bait in their prime spots initially. So comparing organo-bait catch with, say, herring, menhaden, or shad could be challenging if they did not want to put it in the ideal space. We had solutions where we would help them offset their cost during the development phase and also including them along that process. And we learned very early on that they were pleased that very few people ever took us up on that stipend reimbursement. So future initiatives and moving forward, right now we're at the stage where we want to move into commercialization. We're developing at an R&D level, and so we are seeking capital to purchase the raw materials at scale such that our bait technology can compete with those that are currently out there in the industry. We also believe that there are many other opportunities for organobait. We chose the fishing industry first because it seemed lower risk and easier to penetrate. But we do think that some of our chemistry know-how, some of our experience could lend itself well to the aquaculture industry in the future. As we're going to market, we're planning on attacking the recreational segment first because of the higher margins associated with it, and we're working with several groups in Louisiana. Crawfish industry is desperate for a new solution, and it's growing rapidly, and everything is contained within 3 parishes, so we think logistically it'll be easiest for us. Thank you.
I think we've got time for a question if anybody has any. There you go, Lucas.
So I don't only like to grow fish, I also like to go fishing. And so I apologize if anyone had a really serious question, but I saw you had something about ice fishing.
Yeah, my wife's Canadian.
Oh, so it works for ice fishing as well?
We do have terminal tackle embodiments that we have developed, and so we are looking at it as a cross-platform technology. A lot of these chemosensory molecules are relatively hardwired, so we've seen efficacy across different platforms.
Yeah, hi, I liked your presentation. I like what you're doing. What are you looking for in terms of capital, and what are you going to do with it?
Yeah, raw materials is really where we need to be. Right now, we're purchasing our molecules at very small scale, R&D quantities, so we can't compete with the stone crab industry that's using cat food cans as their bait right now. $250,000 would get us to that moderate level scale. $400,000 would allow us to get to probably those 85% margins relatively easy. Also, no one really on the team is a fishing participant, so we would need to have some talent acquisition there. I'm the president and technically the CEO. I'm probably the early stage CEO, so there would be a much better person to take my role where I would do better and excel in the science.
Okay. We'll talk later.
Excellent, thank you.
Thank you very much.